Hi, morning. How are you? Are you okay? Thank you so much. I'm good also. This time I will continue our discussion entitled Lipid Metabolism Part 3. The topic that I will discuss for this time is Cholesterylogenesis and Lipogenesis. Let's see first cholesterol. Cholesterol is a major role in the animal tissue. Cholesterol present in tissue and plasma as free cholesterol or as a sorate form combined with long-chain fatty acid. Consists of 4 fused rings donated by letter A, B, C, and D with branch hydrocarbon chain attached to the D ring. Cholesteryl ester. Most plasma cholesterol is esterified with a fatty acid. Cholesteryl ester are not present in a membrane. Cholesteryl ester present in a small amount in most cell. Let's see this one. This is a cholesterol. This should be hydroxyl group. Then this is a fatty acid tail. Cholesterol and lipoprotein. Let's see this one. It's a nice picture to explain something related to this cholesterol and lipoprotein. In plasma, both free cholesterol and cholesterol ester are transferred in lipoprotein. This is low density lipoprotein. Free cholesterol is removed from tissue by plasma high density lipoprotein and transferred to the liver in reverse cholesterol transport. Let's see this one. The structure of this one. This is phospholipid monolayer. You can see from this picture. This is cholesterol ester. This free cholesterol. Then you have in this picture phospholipid monolayer, and this one is ApoB100. Now let's discuss function of cholesterol. Cholesterol is the most abundant steroid in human body. Several essential functions. Cholesterol has a major constituent of plasma membrane and plasma lipoprotein. Cholesterol is a precursor of bile salt or bile acid. Cholesterol also precursor of steroid hormone, adrenocholesterol, sex hormone, placental hormone, and etc. And cholesterol also precursor of vitamin D and some alkaloid. And cholesterol regulate, the cholesterol required for nerve transmission and abundant in nerve tissue. Now let's see liver cholesterol. The main source of liver cholesterol are dietary cholesterol, cholesterol synthesized extrahepatic in extrahepatic tissue or from de novo synthesis in the liver. Let's see this one. The picture, the right side. This is the major source of liver cholesterol. And from this side, the major road with cholesterol leave the liver. So we can see from this picture, this is dietary cholesterol. Will enter this one and pull in a liver cholesterol. The same thing, this high density lipoprotein will enter this pool. Then from this pool, the cholesterol will secreted by this one. Let's see. You can see this one. Now let's move to cholesterol synthesis. Cholesterol synthesized in all tissue. And major site for synthesis is a liver, adrenal cortex, test, ovary, and intestine. All carbon atoms are derived from acetyl-CoA. Enzymes involved in biosynthesis are partly located in endoplasmic reticulum and partly in cytoplasm. Let's see this one. This is the pool. This is the blood. This is hepatocyte. And this is a bile. Let's see what. So if you look this picture, you can see that this one will enter to the cholesterol pool, and from this cholesterol pool, it can be used to convert this cholesterol into cholesterol ester and to another molecule such as bile acid. Now let's move to synthesis of HMG-CoA. In discussing the synthesis of HMG-CoA, we should have two molecules of acetyl-CoA will be condensed into acetyl-CoA in a cytosol. Next, cytosol HMG-CoA synthase will add third molecule of acetyl-CoA to acetyl-CoA to form C-carbon compound HMG-CoA. Let's see this reaction one by one and step by step. Have a look this one. We can see the mechanism of joining this small molecule into large molecule. Cyclus. Then let's move to synthesis of mevalonic acid. HMG-CoA reductase is the regulatory enzyme in the next step. This enzyme reduces HMG-CoA HMG-CoA to mevalonic. HMG-CoA reductase is endoplasmic reticulum membrane enzyme with catalytic unit hanging in the cytosol. And synthesis of malonate is the rate limiting and key step in cholesterol synthesis. Let's see this one. This is 3-hydroxyl 3-methyl-glutaryl-CoA will convert it into this mevalonic acid by catalyzing with the enzyme HMG-CoA reductase, and this reaction probably will occur by oxidizing this NADPH to NADP+. Now, have a look to cholesterol synthesis. Production of 5-carbon unit isopentenyl pyrophosphate. We call this one as EPP. Then this EPP will be condensed through several steps, result in sequelant. This sequelant consists of 30 carbon. Then cyclization of a sequelant will produce lanosterol. Lanosterol consists of 30 carbon and will convert into cholesterol with 27 of carbon. Let's see this pathway. We have this acetyl-CoA. This acetyl-CoA will convert into this HMG-CoA. Then from this starting point, that will convert to a sequelant, and by cyclization procedure, this sequelants will convert into lanosterol. Then this lanosterol will be converted into cholesterol. Now let's see cholesterol biosynthesis pathway. The same thing like we discussed before, that this biosynthesis will start from acetyl-CoA, then move to HMG-CoA, and will be converted into mevalonic. From mevalonic will convert into farnesyl pyrophosphate, and this is starting material. The important starting material from this farnesyl pyrophosphate can be converted into this one, and also converted into a sequelant. Then from this sequelant, by cyclization procedure, will be converted into cholesterol. Then again, from this farnesyl pyrophosphate, it can be converted to farnesylated protein. Then from this farnesyl pyrophosphate also can be generated ubiquinone and granulated protein. So this is the biosynthesis of cholesterol, or cholesterol biosynthetic pathway. Then let's move to the regulation of this cholesterol synthesis. Have a look to this picture in the right side. This is the pathway, and you have in this part cholesterol, and you have also in this part again, and from this side you have insulin and other molecule, and also ending story of this one will be cholesterol. Then let's see, HMG-CoA reductase is a feedback regulation of the transcription gene activated by the sterol regulatory element binding protein. And insulin and thyroid. Insulin promote the phosphorylation and favoring cholesterol synthesis. Thyroid will stimulate the LDL receptor to break down and remove LDL. Now let's see the hypercholesterolemia. Hypercholesterolemia is a condition that the level of cholesterol is high. So we can say that high level of cholesterol concentration in the blood, that hypercholesterolemia, and this lead to arteriosclerosis. This hypercholesterolemia can treat using statin drug, the structure analog of HMG-CoA. This is a competitive inhibition to HMG-CoA reductase. Let's see this one. This is a vessel. This is the plaque. This is the problem of making arteriosclerosis. And then we can see from this one, how come that this statin can be used as a drug? Because this statin will block this enzyme. This is a competitive inhibition. Let's see this one. So I think for older people, it's not new for them to understand what statin is. Now let's move to lipogenesis. Lipogenesis is an anabolic pathway by which fatty acids are synthesized from acetyl-CoA. Not simple, a reversible of the beta oxidation pathway. Let's see the difference between beta oxidation and lipogenesis. Difference on like reaction site, enzyme involved in the reaction, intermediate carrier, coenzyme, and carbon atom. Let's see this one. A beta oxidation occur in a mitochondria, and fatty acid synthesis occur in a cytosol. Enzyme involved in this reaction, according to the reaction, for fatty acid synthesis should be fatty acid synthase, and a coenzyme involved in the beta oxidation are FDA and NAD+, but for fatty acid synthesis is NADPH, and a carbon atom remove 2 at a time, but for fatty acid synthesis add 2 at the time, completely different. Now let's see a Citrate-Malate Shuttle. This is a shuttle, shuttle. This is mitochondrion, and this is a cytosol. This shuttle can enter this cytosol by this shuttle. But have a look to the down side of this picture. This pyruvate from cytosol can enter mitochondrion through this shuttle. That's the acetyl-CoA is the starting material used for lipogenesis. Acetyl-CoA generated in mitochondria and should be transported to cytosol for lipogenesis. The movement of acetyl-CoA involves a citrate ion, oxaloacetate, and malate. Then let's see lipogenesis step by step. Let's see. This is the first one, activation of acetyl-CoA. This is acetyl-CoA carboxylation, and then elongation of fatty acid chain and termination of fatty acid synthesis. Now, let's start from acetyl-CoA activation. Catalysis involved in this acetyl-CoA activation is acetyl-CoA carboxylase, and the product of this acetyl-CoA activation is malonyl-CoA. This is irreversible reaction and rate-limiting step in lipogenesis. Acetyl-CoA carboxylase is under allosteric regulation. Citrate is positive effector and palmitoyl-CoA is negative effector. Now have a look to this down side. There is a reaction of this one. You can see that this is acetyl-CoA and this comes from bicarbonate. Then this one will be converted, but it's need energy as ATP and will be converted into malonyl-CoA. Let's see again. This come from acetyl-CoA, and then this part come from bicarbonate. Now let's discuss chain elongation. Have a look this one. We start from acetyl ACP. This is the first one is a condensation, hydrogenation, dehydration, then hydrogenation again. Let's see. Acetyl-CoA used as primer and attached with fatty acid synthesis. Elongation occurs through repeat addition of 2C fragments from malonyl-CoA to the primer. Each process uses 2 and ADP2, consists of condensation, reduction, dehydrogenation, dehydration, and another reduction step. The step of malonyl-CoA pathway occur with the growing chain esterified to acyl carrier protein. We call that one as ACP, acyl carrier protein. Now let's continue to chain elongation. The sequence of cyclic, the sequence of cycle need to produce a long chain fatty acid from acetyl ACP. Malonyl ACP add to carbon at each cycle. Its loop represent one cycle. Again, its loop represent one cycle. Then let's discuss the termination of fatty acid chain synthesis. This termination occurs when 6 carbon in palmitic acid is formed through 7 repeat elongation. So the next reaction of this reaction is A acetyl-CoA plus 7 ATP plus 14 NADPH will convert into palmitic acid plus 7 carbon dioxide plus 14 NADP plus 7 ADP. I think that's all for this moment. Thank you so much again. You are so wonderful. Thank you again. Stay safe and see you again. Thank you.